Hello, seventh grade. Today we are learning about mutations since that does have a lot to do with DNA. But first, let's go ahead and review what we learned about Punnett square ratios from yesterday. What is the ratio of heterozygous to homozygous offspring in a parent crossing of big B, little b, and big B, little b? So let's go ahead and start ourselves off with creating a key. So let's do this. Okay, key. There's a big B and there's a little b. It doesn't even matter like what the phenotype is because they're not even asking of us about phenotype at this point. But I know that big capital letters means dominant and little letters means recessive. So great. And here's our parents. We have parent 1, which is big B, little b, and parent 2, which is big B and little b. Let's go ahead and put that into a Punnett square. Go ahead and figure out what is the first box. That would be big B, big B. What about the second box? That's right, big B and little b. And our third box would also be big B and little b. Go ahead and figure out the fourth box. That would be little b and little b. All right, so let's go ahead and figure out step 4. What do we want to know? So we want to know, first things first, which ones are heterozygous and which ones are homozygous. So let's go ahead and label those on our Punnett square first. So let's look at this first box. Is this heterozygous or homozygous? This would be homozygous. And what about the next box? That would be heterozygous. Third box, also heterozygous. And our last box, also homozygous. So let's go ahead and we have to do 2 ratios here because they're asking for heterozygous and homozygous. So let's find the ratios for heterozygous and homozygous. Heterozygous, we have 2 out of 4 squares. So what's that percentage? Well, actually, we can figure that out later. Homozygous, we have 1, 2 out of 4. Go ahead and do your math and figure out what is the ratio of heterozygous to homozygous offspring. That's right. If you simplify these fractions, they turn out to 50%. So we have 50% heterozygous and 50% homozygous, which would be B. And that is how you would solve this kind of problem with Punnett squares. Let's go ahead and keep going. Today we are going to talk a lot about DNA base pairs. So let's go ahead and practice that before we start off on our mutations lesson. So today your CFU is going to be, what is the base pair to this strand of DNA? So you're going to type it into the box, but first let me get you started with some steps. Step 1, always write. Always trust good cats. And then when I finally write that down, I'm going to draw a line between trust and good so that I know that A matches with T and G matches with C. And now go ahead and match the base pairs. And so just to check on this, it would be G, C, A, C, G, T, T, C, A, G, T. And how I know that is literally just because of this. It tells me that adenine matches up with thymine and guanine matches up with cytosine. So hopefully you got that right and hopefully you checked your work. All right, so why do people have different traits? We all look very different, right? That's because people have very different base sequences. Their DNA, oh my goodness, stop. Their DNA looks very, very different from the next person. Different base sequences result in different types of proteins, which result in different types of physical traits. So what this is saying is like Somebody might have their DNA coded for GCATTA, and somebody might have it coded as GTACCG. Those are different, right? So they're going to come out with different traits. This person might have brown eyes, and this person might have blue eyes, because these code for different genotypes. So beneath the genotype, which is already a code, lies even more DNA code at the basis of it. And a lot of different mutations can happen to these DNA sequences. So what is a mutation? A mutation is not just Teenage Mutant Ninja Turtles, not just X-Men. A mutation is when a gene is changed. The structure of a gene has changed. It's mutated. The result is when you mess with the structure of a gene, there's a bunch of different codes that you can mess up, and then that is transmitted to different generations. So like if your parents had a mutation, they might have passed that down onto you, and you might pass that down on to other people. Let me just say this before we continue with the rest of the lesson. Not all mutations are bad. In fact, a lot of mutations have helped humans and other species evolve and become better versions as you go through time. But we'll get to that. So what this diagram is basically showing me is that with DNA, you'll recognize these as base pairs. Sometimes you might have an incorrect copy, and when that copy is incorrect, it turns out to be mutated. There's actually really specific ways that DNA can be mutated. We're going to go over 4 different ways that DNA can be mutated today. Oops, there it is. So what are the, what is that? What are the different types of mutations? We have 4 different types of mutations: substitution, insertion, deletion, and silent mutations. We're going to go over all of these in a little bit, but substitution is basically if you substitute the right base pair for the wrong base pair. Insertion is when you accidentally add a base pair. Deletion is when you accidentally delete one. Silent is when nothing happens, which is often the case. So let's just go over them one by one. Substitution mutation, sometimes we call that a point mutation, but we're going to call it substitution mutation for this class, is a type of mutation when you replace a single base with another base. So for example, here we have CTT, and when we write always trust good cats, you would know that C matches with G, T matches with A, and T matches with A. But when you substitute mutate, that means that you replaced a pair wrongly. So you accidentally changed a pair and it doesn't turn out right. So for example, oh, I'm so sorry, I totally said that wrong. Basically is what it's saying is that if you accidentally sub out a certain base pair. So for example, here we had CTT, and that's right, but what if they accidentally write CAT? That would be wrong, because then it would make a completely different, it would just change a lot of things in a bunch of weird ways. So what we're basically saying is that a substitution mutation is when we accidentally change something that's not supposed to be there. So for example, in this example, we have CGAATTG. Substitution mutation might just randomly change it from CGTATTG. And that might mess things up along the way. Even the slightest mistakes in passing on DNA could change an individual very drastically. So that's substitution mutation. You replace it with something that's wrong. There it is. All right, the next type of mutation that we have is insertion. And insertion is exactly what it sounds like. It's when you add or you insert a base pair, like a random, like little what are they called? Like random little base pair into a DNA sequence. So here in this diagram, you can see this is a DNA strand that's perfectly normal, all great. But then all of a sudden, in between these two guanines, you accidentally insert a random adenine. You added a random, like, little nucleotide. And that's not good because then that shifts the whole sequence. Now everything behind it is completely off. So this one's a little bit more drastic because we're going to insert a random, like, nucleotide and just put it in there when it really like just doesn't belong there. So for example, in this sequence, CGA, everything is going normal. Then all of a sudden I'm going to add a G and continue on. Here we see that it messed up because you added or you inserted something that wasn't there before, something that wasn't supposed to be there. So that's insertion. The third type of mutation is deletion. Now you can probably like infer what that means. A deletion is a mutation when a part of a DNA sequence just gets deleted. It just gets yeeted out of the way, which means that you like lose some of the material. So in insertion, you added a DNA or a little nucleotide. In deletion, you're going to take one out, and that's also going to throw off the rest of the sequence as well. So for example, in this example, let's see what they did. Oh, here it is. They deleted the C out of the original DNA strand, and now the G shifted, and now everything kind of shifted. And so everything got a little bit like messed up when we took out this C. You deleted it. And that would cause a type of mutation. Last but not least is a silent mutation. So silent mutation just means that sometimes the DNA gets a little bit messed up, but it doesn't really do anything. So for example, if you change the A to a G in this situation, it just didn't really mess anything up, and your problem will tell you when it messes something up or when it doesn't mess something up. So if perfectly everything else goes perfectly fine, that is called a silent mutation because it didn't really like do anything. So let's go ahead and try a couple practice problems. What I like to do is I like to write SID at the top, just to remind myself that the options are substitution, insertion, and deletion. So I can either substitute or change part of the DNA sequence, I can insert a random nucleotide, or I can delete one. So let's look at this original piece of DNA, TCACCG. And then we see that it got mutated to TCAACCG. Now I'm gonna like kind of look at like what has changed here. So this is the same, this is the same, this is the same, this is not the same, but this is the same, and this is the same. So what it looks like is that they inserted that random A into the middle of everything, and I can tell that they added something because everything shifted that way. So because like you literally have to just like look at it and like try to notice what has changed. And in this case, that would be insertion. Now go ahead and try this next one by yourself. The answer to this is that it is substitution. Because there's the same number of DNA sequences, 1, 2, 3, 4, 5, 6. You didn't take anything out, you didn't add anything. The only thing that changes is that like the A randomly changes into a G. And when that change occurs, That would be called a substitution. Now try the next one. All right, so we did see that there was a substitution over here, but my problem tells me that it codes for the same proteins. If it tells me that it codes for the same thing, that means that nothing really changes. Everything is fine, everything still turned out the same way. So since everything turned out the same way, this would be silent. And then last but not least, go ahead and try the last one. I can see that there was a subtraction over here. You have one less from the DNA sequence. And if I check, it looks like they deleted this A. They took it out, so that would be delete. And those are 4 types of mutations. So now go ahead and we're gonna try another one together. The original sequence is CACCTT. The mutated sequence is CATCTT. Number 1 is to circle the change in mutated DNA. I'm actually going to circle it for you because you can't circle it. You don't have the paper. And so this is the same. This is the same. I see a weird change over here. This is the same. This is the same. And this is the same. What kind of mutation is that? Yup, that's substitution because it just randomly changed. Now, try the next one. I'm going to circle the mutation in the DNA. So T and T are the same, G and G are the same. There's a discrepancy here. Now there's one here. This one's the same. So go ahead and try to think about, oh, and then this one is missing one. So go ahead and try to think about what went wrong here. As you can see, they deleted the A that was supposed to go in between the 2 G's. So this one is deletion. And that's pretty much it for practice today. I just wanted to end the lesson by saying that not all mutations are bad. So a lot of us, like, when you hear mutation, you think like, oh, this person had a mutation, clearly like their life is horrible and like everything went wrong. Totally not true. Some mutations are actually pretty good. So like some people actually have like this mutation within the DNA that codes for cholesterol levels in their blood cells. So some people actually have this mutation that makes them have less cholesterol, which means that they have less chance of heart disease, which means that they have a healthier circulatory system. So that's good. Another like really common mutation that a lot of humans have, but a lot of humans don't have, is lactose tolerance. So originally, like when humans like were first made, we weren't like really meant to drink cow milk. That's milk for cows, not milk for humans. But over time, since humans domesticated cows and we lived with them and we farmed with them and we used them often, a lot of humans started to develop a mutation that would allow them to successfully digest lactose from cows. So if you have this mutation, you are good to go when you eat ice cream, when you drink milk, nothing horrible happens to you. But Miss Aquino does not have this mutation. I have lactose intolerance. So I personally cannot drink a lot of milk or else I'm going to have a bad time. So some mutations are good for humans. And that's the end of today's lesson. I hope you guys got it. You will have more practice in your worksheet and before the test. So text me if you have any questions.